Let us come to finish the replication, how the replication is finished. So completion of a DNA replication requires a set of specific events, and these events are different for circular chromosome and linear chromosomes, or you can see these events are you can say these events are different in prokaryotic cells and eukaryotic cells. In case of circular chromosome, the conventional replication fork machinery replicates the entire molecule, but the resulting daughter molecules are topologically linked to each other. So, in a circular chromosome, replication will proceed, and at the end of replication, the two chromosomes will be linked to each other. As you can see here in this case, so in this case, this is a circular DNA chromosome. It starts replication from here, and then replication proceeds in both the directions, and as a result two chromosomes are formed, but these two chromosomes are linked to each other at a specific site. So this is a problem of circular chromosomes. While in case of linear chromosomes, the problem is different. The replication machinery cannot completely complete the replication of very ends of the linear chromosomes. So these very ends of the chromosomes are called telomeres. So as you have seen in when the replication will complete, the ends which are called telomeres, they will pose some problems. So the replication machinery cannot complete replication at the ends in case of linear chromosomes. Therefore, organisms containing linear chromosomes have developed novel strategies to replicate their chromosome ends or telomeres. So come to the circular chromosomes. After replication of a circular chromosome is complete, the resulting daughter DNA molecule remain linked together as a catenanes. Catenanes are just like two circles joined in this way. As this is a single chromosome, but at the end these chromosomes will be joined in this way. So these form catenanes. So in this way these two are linked together. So to segregate these chromosomes into separate daughter cells, the two circular DNA molecules must be disengaged from each other, or this is called decatenated. They are catenated and they must be decatenated so that one chromosome goes to one cell. This separation, this decatenation, is accomplished by the action of type 2 topoisomerases. So these are different from topoisomerases which you have seen. So the action of type 2 topoisomerases that results in the decatenation of circular chromosomes.